Okay, here's your review from Monday. It's Wednesday, I think. I'm not sure. Pretty sure. Okay, 30 more seconds. Okay, first one. What is the correct way to write a scientific name using binomial nomenclature?
Genus and species with
What else?
lowercase.
Capital genus, lowercase species, and underlined or italics, depending on how you're doing it. Good. I'm emphasizing this with you so that when you write things like E. coli in a lab report, you'll do this. Okay, what is one reason that virus is not considered to be alive? Shauna.
It can't metabolize
to live, to metabolize, and
not reproduce.
to reproduce. Okay, so they're really good at doing those things as long as something is helping them. They are obligate intracellular parasites. Are they alive or not? Still think there's some debate. Because we talk about killing them. But if they're not alive, how do we kill them? I don't know. What are the two parts of a virus? Sometimes there's more than two, but there's two that are of all of them here. The capsid, so that protein coat, and then the genetic material, which can be a variety of different things. And then what kind of virus infects bacteria? There's only one kind that does. Bacteriophages, which we sometimes just call phages because we're lazy. We don't want to say all those syllables. Good. Just a couple of reminders before we dive into chapter 19 a little bit more. Lab this week is happening. You have a quiz this week. This will be your first lab quiz. It will be covering what we talked about last week and what we are doing this week in lab. Pre-lab 2 is due when you get to lab, like when you walk in the door. We are going to meet in 210 again, not our normal lab, but we're going to meet in 210 again because we're using bacteria, and we can, because next week we'll go back to our normal lab, but today we'll be in 210. I did move the folders, the crate of folders into 210, so if there are papers that you're getting back, which you all should have something, they will be in 210 with us today. I feel like there's one more thing and I don't know what it was about lab. Nothing about it. Oh, there's something in lab I know that talks about the SWI microbes. We didn't do that last semester, so we can't really continue that this semester, so you can ignore anything that talks about SWI microbes. Sorry, I don't even remember if there's a question in the pre-lab about it. If there is, I'm sorry. I was debating whether to pull some microbes out of the freezer and decided it wasn't worth it because we haven't done the other stuff to get to that point. So we are not doing SWI microbes stuff at all. Okay? In terms of lecture, just a reminder that next Friday, not 2 days from now, but a week from 2 days from now, the 4th of February is going to be your first test. It's going to cover chapter 18, chapter 19, what we're covering, a little bit of chapter 26, chapter 27, which we're going to start today, and chapter 28, which we'll cover next week. Okay, that's the other thing. Outlines. I started grading the ones online, so some of you have a grade, some of you don't yet. Sorry, I'm getting there. And those of you who gave me hard copies, I'll put those in your folder in lab, hopefully today. For outlines for the future, at least as long as I'm here, for February break, until February break, I don't care if you give me a hard copy or turn it in online. Either way is okay with me. So some of you I know are like taking pictures of yours and like figuring out how to upload pictures. And if that's more of a pain than just scanning it and printing a copy for me, that's okay. So whichever way you want to turn it in is okay with me. If you would prefer to give me a hard copy, I like that. If you would prefer to turn it in online, that's also fine. They're not that hard to grade online. Okay? So your next outline I think is due Monday. Katie.
What time?
I think it's class time. I think that's what I put.
Knowing the chapter 28 outline will be good.
I think I might have changed that yesterday to the following Monday.
The Monday after the exam?
Yes. Would it do you good to get it done before the exam? Yes. But I'm not going to make it due on the day of the exam. So I think the chapter 28 outline, I don't know if I might not have put it on there yet, but that would be the 7th of February? Yeah, the 7th.
Just to make sure, so the test was the Friday, February 4th?
Yes.
Okay.
The test is Friday, February 4th. And that's on Moodle, and I put the information. And remember when you look at the syllabus, when it talks about different chapters, sometimes it says read certain sections, and not all of them. I'm not going to test you on the sections that you don't have to read. And for chapter 18, we're only testing on those first 2 sections, that's on Moodle. Okay? So anticipate today we'll be able to finish chapter 19 without a problem. We're going to start into chapter 27. So there's a big skip there. We did a little bit of 26. We're going to start in chapter 27, which is a survey of prokaryotes, bacteria, and archaea. And then we'll start chapter 28 next week, which are the forests. My husband must be trying to sign into Dropbox because I just got a message with a code on it. Okay. This is not an exhaustive list. Actually, let's just go back for 1 second. So just as a reminder, we talked about, and you need to be familiar with, the overall replication process for a virus. This again is a generalized one. This is a generalized process that we talked about. And then we also talked about the differences with an enveloped virus. The envelope has those glycoproteins and that lipid membrane that help it to bind to a host cell better. And then the host cell has to donate part of its membranes generously to make more little viruses to infect itself. So this is not an exhaustive list, and it's a color-coded list, because I like color coding, of viral infections that are common to humans. I would guess you've heard of most of these. Some of them are more famous than others, and some of them have had waves of famousness and waves of not famousness. So I color-coded them because there are ways that we categorize viruses. Remember we talked about the fact that they can have different kinds of genetic material. What are the two different kinds of genetic material? DNA and RNA. And there's double-stranded DNA, single-stranded DNA, double-stranded RNA, and single-stranded RNA. We're going to talk only about double-stranded RNA and single-stranded RNA and double-stranded DNA viruses. So I color-coded them so we could talk about the categories. Again, some of these you're familiar with, some of these you've probably had, and some of these you probably are vaccinated against and won't have, and some of these you will hopefully never, ever, ever come in contact with, even because you don't want to have them or think about them. Like Ebola freaks me out a little bit. Oh, sorry, I forgot to hit the writing button. Okay, so these red ones, influenza, measles, mumps, and rabies. I did not put these in order of letters. It just happened, in numbers. This is the way they fit on the slide. These are single-stranded RNA viruses. They are category 5 viruses. So all of these have a single strand of RNA. What puts them into category 5, there's 3 different categories of single-stranded RNA viruses. What puts them into category 5 is they're what we call a negative sense virus. So that means when that genetic material from the virus enters the host cell, it can't be directly translated. So negative sense refers to the fact that it cannot be directly translated into proteins. The cell actually, the virus brings its own RNA polymerase to transcribe it into a readable mRNA. So that RNA polymerase makes when we say readable, we mean translatable. When we say readable, we don't mean my handwriting. So that viral RNA polymerase will translate that viral RNA into a readable mRNA so that the cell can start making proteins for the virus. It's so nice of the virus to bring its own RNA polymerase to the party. So it's category 5, it's a negative sense RNA, so it can't be directly translated. Does that make sense? Negative sense or positive sense? Negative sense or viral humor is not that funny. Okay, the next one, human immunodeficiency virus, you've probably heard of. Anyone know what makes this one super special? It's also a single-strand RNA. So it's a single-strand RNA virus, but it's a different color, so that tells you it's a different category. It's a category 6. So it has a positive sense RNA, which generally means it can be directly translated, but it's not. It also brings its own enzyme to the party. Instead, the RNA is transcribed into DNA. Why is that weird? It's backwards. Okay, we usually do DNA to RNA. We're doing RNA to DNA. So the enzyme that does this is called reverse transcriptase. There's an S in there. So transcriptase, ACE tells us it's an enzyme, transcript tells us it's doing transcription, and reverse tells us it's doing it backwards. So it's taking the viral RNA and making DNA out of it, which is then the cell will transcribe and translate to make proteins for the virus. It's kind of in a category all by itself there. It's the only single-strand RNA category 6 virus that we'll talk about. The next two, again, you've probably heard of as well. Herpes virus. If you've ever had a cold sore.
What is the difference between category 5 and category 6? What determines that?
So category 5, the RNA is going to be it's not even really transcribed. The RNA is going to be copied by our viral RNA polymerase into mRNA. In category 6, the RNA is transcribed into DNA and then translated by the cell, transcribed and translated by the cell into proteins.
So it's basically an RNA version of
Positive sense means that the RNA could be directly read, but in the case of HIV, it's not directly read because it's turned into DNA and then read from that. So it's directly read into DNA, I guess. It's not directly read into proteins. And actually, let's skip herpes first. Let's go over here to all the orange guys, because they're all in a category by themselves. And these are the last of our single-strand RNA viruses. They're single-strand RNA virus category 4. And these are positive sense ones. So positive sense meaning they're directly translated. So the viral RNA is directly translated into proteins. There's no intermediary steps. We don't have to make another RNA that's complementary but readable. We don't have to make DNA. It's direct transcription into direct translation, Direct translation. It's what? Category 4, yep. So that includes things like West Nile virus. What do you know about West Nile virus? Who carries it? Mosquitoes. That's a mosquito-borne virus. Ebola hemorrhagic fever. It's just terrifying. Anybody seen, what is the movie? Outbreak? I think it's Outbreak. There's a movie Outbreak that's about Ebola. There's a book that Outbreak was based on that I think it's called The Hot Zone. I was like, you can see it on my shelf. It's called The Hot Zone. That talks about Ebola cases that were discovered in monkeys that were used for research in the United States before we really understood anything about Ebola. It's terrifying. Okay, and then you might remember there was an Ebola outbreak 7 or 8 years ago, I think. Like was the first of there's continue to be Ebola outbreaks, but there was the first of it. It was also terrifying. Ebola is just terrifying. Hepatitis A through E are a category of viral illnesses that affect what organ? The liver. Anytime you see anything with hepa or hepato in it, that's going to be liver. So there's a bunch of different categories of it. There's different modes of transmission for all of them, different symptoms, that kind of thing, but they all are liver issues. Dengue fever and yellow fever are tropical illnesses more. So if you've ever traveled to a more tropical country for an extended period of time, you might actually get a vaccination for yellow fever or dengue fever. You have to work kind of hard to get them because they're not ones you can just go to the doctor because the doctor doesn't have them sitting around because we don't worry about getting yellow fever in 10-degree weather. But if you're going to travel to a tropical country, you might, or in an area that's at risk for yellow fever, you might get those vaccinations. Zika fever was kind of a big deal, like 5 or 6 years ago. Do you remember that? Zika can cause significant birth defects, so it was especially a concern for pregnant women. And then coronavirus, I don't think we talked about the right coronavirus, maybe more than we want to be, and maybe not as scientifically as we want to be. And then rhinovirus that causes colds. So all of those are positive sense single-strand RNA viruses that can be directly translated into proteins. Their genetic material is directly translated. And norovirus, you'll see over here, is also orange. It's in the same category. Norovirus and rotavirus are on the same line because they cause similar symptoms. What are those? What are the symptoms of those two? Maybe you've never experienced them. I hope you've never experienced them. Norovirus and rotavirus. Significant projectile vomiting. Last semester, those of you who heard me watch Vomiting Larry? Oh, that was micro. In your free time, look up Vomiting Larry. Vomiting Larry is a model used to model how viruses are spread through projectile vomiting. Somebody invented it because that's what they do. Scientists do in their free time, not in their free time, in their days. So norovirus and rotavirus are both gastrointestinal viruses that cause significant vomiting and diarrhea. We had the plague of norovirus 5 or 6 years ago. Minor Hall was just like a disaster. From what I heard, I wouldn't have gone in there if they'd cleaned me. I would not have gone in there. It was just everybody was vomiting all the time, and like couldn't get to places that were appropriate to vomit. And I heard Minor Hall was just like covered in vomit. That's not to say girls were unaffected by it, but it seemed that Minor Hall was more affected than girls. I'm not making any comments on hygiene, but yes.
So the norovirus would be category 4.
Yes. Rotavirus is, let me check my notes because I don't remember now. Rotavirus is a double-stranded RNA. So it has an RNA with a complementary RNA strand, which is weird because RNA is single-stranded generally. Rotavirus, you're actually vaccinated against as a child. It's an oral vaccine, which I only know because I take my children and you take them when they're little teeny babies and they try to get them to drink stuff and I'm like, and it's all running out their mouth. They're like, it's fine, they got enough. I'm like, did they? Because I don't want to vomit, so give them some more. I hate vomiting illnesses. Herpes virus is cold sores, but also has some other sexually transmitted disease consequences. There's different herpes simplex virus 1 and herpes simplex virus 2. And then varicella zoster, what is that one? Most of you probably have never experienced it because you were vaccinated against it. But I was not because I'm too old.
Chickenpox.
It's chickenpox.
Really?
Yep. So both herpes virus and varicella zoster are double-stranded DNA ones.
Did you get the pox?
I got the pox.
How the pox do you get shingles now or no?
I could still get shingles.
Isn't there like a separate vaccine?
There is a separate vaccine for shingles, and I would happily get that vaccine because chickenpox is bad.
Wait,
and I've heard shingles is
how
worse.
painful is chickenpox?
What?
How painful is chickenpox?
How painful is it? I was in 2nd grade. I don't know. I was itchy. I was miserable. They got infected, so I had to take antibiotics. I had to stay with my neighbor because my parents were working.
It's pretty much the flu with mosquito bites.
Well, they're all over your body. I was 7. I don't really remember a whole thing. I remember that I had to take yellow medicine that tasted bad because they got infected and they were itchy. And my parents actually think I got them twice. They think I had it as an infant. I had like 2 or 3, but then I got them again when I was 7.
Did you not get it again if you already had them?
Nobody are double-stranded DNA ones. So those have DNA in them. Okay, so those of you who continue on to microbiology will spend more time talking in intense detail about symptoms and discussing characteristics of viruses. Yes, Athena. Whoops.
Are there any single-stranded DNA ones that we know about?
No.
Okay.
I can't even give you an example of a single-stranded DNA one. That's how much viral knowledge I have.
Does the positive sense versus negative sense like increase its ability to infect or stay in the body?
Not that I know of.
Because like the single-stranded positive sense, those are all ones that for the most part have quick onsets, right?
Single-stranded positive sense.
Like except for like hepatitis.
I would say yes, those are all pretty, but rotavirus and norovirus are also pretty quick onset. Flu can be quick onset. Measles and mumps can be too. Rabies, I forget what the incubation period is. I'd have to look it up to tell you anything definitively.
Yeah.
Some of them, like herpes, also has the ability to remain latent. So like once you have herpes in your body, it's always with you, and it comes back in times of stress. Dr. Smithers, I don't think would mind me telling you, she was infected with herpes simplex virus 1, and before masks came out, we would know when she was feeling stressed because she would get a cold sore, and you could always tell.
Wouldn't you always say she had her DNA from the
Yes, she would say she was a genetically modified organism.
So the double-stranded, like they're not categorized.
They're not categorized because double-stranded DNA is just double-stranded DNA. There's no categories. It's just a single-stranded RNA because the cell deals with it differently. Okay, this is a picture of COVID-19 SARS-CoV-2. Again, because we're spending a lot of time with this virus, it's not bad for you to know. It's an enveloped virus. So what do we know about an enveloped virus? It's going to have glycoproteins that will allow it to bind to the whole cell to enter into the cell pretty readily, and then our cells have to make more of it for them. It's a positive sense RNA. It's a category 4. Positive sense single-stranded RNA virus. The spike proteins that are found on the outside of COVID target the ACE2 cells of the body. Did I write that down anywhere? No, I did not. So I can't even remember, honestly, because I read about this 4 days ago. I don't even remember what ACE2 cells do. This is bad. I'm not a cell biologist. I'll get back to you on it. I'll tell you in lab. Symptoms of COVID. This is an old slide. So Dr. Smithers made this slide 2 years ago. Like back in the day.
Is this when this began, or is this prior to?
Well, let me think. I'm trying to think. I said 2 years ago, but 2 years ago was no. This is probably from last year. This slide is probably from last year. Symptoms: fever, cough, chest pain. Might have been some more symptoms that are more common with earlier variants. Right now, I just heard this morning, 99 to 99.5% of the COVID positive cases that we're seeing now are the Omicron variant, which the symptoms are a little bit different. I've heard not as much respiratory, sore throat.
Is it good difference?
I've heard it's different. I haven't had it, but scratchy throat, very sore throat, not as much with the cough. It seems to not be affecting the lower respiratory system as much, and more GI stuff. A lot of people I've heard have had more GI issues with the Omicron variant. Again, variants can happen very quickly in viruses because they have small amounts of RNA and DNA, and they reproduce very quickly. So mutations can happen regularly, which we'll talk about with bacteria. So there's always going to be another variant on the way. The immunopathogenesis of earlier variants, at least of COVID, was a cytokine storm, which is when your body, it's an inflammatory response of your body to an invasion. So your body's producing cytokines which cause inflammation. And so one of the more serious consequences of COVID when people get long COVID is called MISC or MISA, depending on if you're an adult or a child, but multi-inflammatory syndrome, where there's inflammatory response throughout the body. MISC is in children and MISA is in adults. It's multi-inflammatory syndrome of children or multi-inflammatory syndrome of adults. But it's caused by that cytokine storm of your body trying to fight it off and causing massive inflammation.
So like some of like the athletes they've made go through like different testing and stuff for like myocarditis. Does that like is that an after effect of all of the different variants or just like one or two?
I don't know about Omicron, but the other ones, yes. That's a concern of long-term effects is myocarditis. So I have a friend who has a heart baby. He was born with half a heart. And so they've been extremely concerned because if he got myocarditis as a result of getting COVID, it might be more than his body could handle because he's only got half a heart. But that's a concern for even healthy athletic people. Myocarditis, a heart infection, inflammation around the heart. And again, I'd say this is an older chart looking at some of the percentage of cases. This is from, it tells us, 2020. So yeah, this is old. But looking at some of the different symptoms and percentages of cases and age risks that some of these different symptoms are seen in the incubation period. Which again, I told my environmental science class yesterday, I've heard a lot of people being upset about science changing its mind. Okay, science does change its mind. Because we learn more things, we have to change our mind. So I looked at this table and I was like, I tried to find a more updated one because I don't know that we'd agree with all of these things because this is from 2020 when we were still in the throes of learning about it. I heard this morning, 2 years ago today was the day that China started like mass evacuations because of COVID and like lockdowns. It was 2 years ago today. 2 years ago today in the United States, I don't think I'd even heard of COVID. I remember like in March, the week before we shut down, they were like, hey, we might go online next week. And I was like, yeah, right, whatever. I won't say that ever again.
What is round glass opaque?
I don't know if that's something in the eye. That's the only thing that I can think of. Something related to a lens of the eye. I didn't look that up because I was too busy looking for any table. I didn't look that up. So that's a good question. And I will look that up for you if you don't look it up yourself.
The amount of times you said, I'm going to look that up in this lecture long.
I'm not a viral expert. Okay. That is the end of chapter 19. Which means we are ready to start with chapter 27, which will take me a second.
Okay, it says on the outline, two viral infections.
Yeah. On the outline? Yes.
Okay.
Did I download this one? No. Oh, good. I forgot to download this chapter. I'm doing well today. Were you all in the building for the fire alarm? Some people were in the building for fire alarm. It threw me off. And I forgot to download the next PowerPoint. Okay, so we are transitioning from things that are not quite alive but have some of the characteristics of living things to things that are most definitely alive and have all the characteristics of living things. So remember on Monday we talked about 3 different domains. When we were looking at classification, we said there's the domain eukarya, which is eukaryotic cells, and then there's the domain prokarya, which is bacteria and archaea. So this chapter is covering bacteria and archaea, both domains. All in one chapter. Chapter 28, when we get into protists, will already be into eukaryotes. So these are prokaryotic cells.
How do you pronounce the second word?
Archaea. My daughter is learning to read, and I'm remembering remembering how many words are not spelled the way that you think they should be. Okay, I don't know how you feel about bacteria, but I love bacteria, and I would like you to love bacteria. So I think when we talk about bacteria, and even viruses to a degree, we always talk about the bad ones, and the things that make us sick, the things that are bad. There are lots and lots, in fact the majority of bacteria are not harmful, and are in a lot of ways helpful to our lives. So I would like you to leave this chapter with an appreciation for bacteria, and bacteria are good for the most part. Again, if you take microbiology, you will not spend much time talking about good bacteria because we tend to focus on this bacteria does this and this one does that and this is bad, 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 bad. Bacteria are good, 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 good. Okay, you're very dependent on them. They do a lot, not just for you personally, but in giant ecosystems. They're really important in nutrient cycling. They're really important in nutrient cycling in your body. Digestion, you couldn't do what you do without bacteria. There are more bacterial cells in your body than there are human cells in your body. Some people say up to a 10-to-1 ratio, which kind of freaks you out. And yeah, it is. You might have 10 times as many. That's not saying 10 times as much mass of bacteria, but sheer number of cells. You might have 10 times as many bacterial cells in your body as you do human cells. What does that mean for you? Are you a human? Or are you a bacteria? A colony. Yes, are you a chimera? Are you both? Are you a hybrid? What are you? If you ever want a book to read about bacteria in you, I've got books to read about that. So these bacteria that are on in the picture are not bacteria that live in your body. This is a picture, I think. I have conflicting, I didn't Google search, reverse search the image. That's like reverse transcriptase there. I didn't reverse search this image, but I didn't image search this, but Dr. Smithers had in her notes that this is the salty lagoon in Spain. Your book has a picture of the Great Salt Lake in Utah. Either way, this is a really salty body of water. Okay, the Great Salt Lake and the salty lagoon in Spain are almost as salty as ocean water. They can approach 32% salinity. And they're pink because of bacteria, because bacteria are amazing. Okay, not only are they prolific in your body and in the world, and we don't even appreciate them, they're also extremely diverse and can do all kinds of crazy things. I think your book uses the term master of adaptation, not so much they can do so many crazy things. But they are adapted to all kinds of extreme environments and can grow in all kinds of crazy places like this lagoon. So this lagoon, or Great Salt Lake, whichever one it is, can turn pink based on the growth of various species, both archaea and bacteria themselves. The bacteria that grow here are what we call halotolerant or halophilic. Oh, I forgot to push the button. Anna, I'm listening. I'm listening.
Are they harmful or passive bacteria?
These I don't think are harmful. There are some bacteria, if you've heard about like red tides or even algal blooms, which those are eukaryotic cells, that can be harmful.
So like,
I don't think these are.
like let's say I consume the water, so
I
I'm
don't-
not gonna die?
I don't think you would die. I don't know that you would want to consume this water. It's really salty. It's like drinking the ocean. I mean, it's as salty as the ocean. So the bacteria that are able to grow in this kind of environment are halotolerant, meaning they can tolerate high salt content. Some of them are actually halophilic, meaning they like high salt. They prefer to grow in high salt environments. So the genus name for them is Halobacterium. Not a very creative name. And I'm underlining it. And this is a picture of them. I could have just underlined it there. They actually have a red pigment in their membrane, which can help them absorb energy from light. So that's why they have it. They don't just have it to look pretty. Why is it an advantage for them to be able to grow in a really high salt environment? Why is that an advantage for them?
There's a lot of space.
Why is there a lot of space?
Organisms can't do that.
Other people can't do it, so it reduces competition, so it gives them an environment to live in that they're not having to fight off other people. Bacteria, not really people. Okay, so when we talk about them being masters of adaptation, there are species that can grow in really high salt environments. There are species that can grow in really extreme temperatures, really hot. If you've ever seen pictures of the hot springs in Yellowstone, they have like oranges and yellows and stuff in it. That's bacteria or archaea. They can grow in really low temperatures. There's bacteria in glaciers. There's bacteria in the Antarctic ice sheets. They can grow in really low pHs. I think your book talks about one that can grow in a pH of 0.03, which is such a strong acid I can't even imagine. But it's adapted to be able to grow in that environment, and nobody else is trying to grow there. So they're not having to fight anything off. They have moisture. They can adapt to lots of levels of moisture, drought, radiation. Some bacteria can survive radiation at really high levels, and they can metabolize lots of different chemicals. There are some bacteria that can break down oil. There are bacteria that can break down plastics. Again, not a lot of bacteria even competing to be able to break down plastics. Nobody wants to eat the plastic. So if there are bacterial species that can eat the plastic, we've got plenty of plastic for them to eat.
Wasn't there a time where some scientists were doing that, but they sent bacteria through the mail?
Anthrax?
Yes.
Yes, there was a time. Right around 9/11, when somebody started sending anthrax, and there was concerns about bioterrorism and that they were related. Spores, we'll talk about it. I think halotolerant more tolerate it and can grow there, but wouldn't necessarily prefer it. Halophilic love it. They love it. So again, just thinking about the diversity of bacteria and their ability to survive in so many conditions, I think they're pretty cool. Okay, so I just said they're really diverse, but let's talk about the prokaryotic characteristics of all prokaryotic cells that they have in common. Some of these you already know, so you can tell me. What do prokaryotic cells have in common? Yes, because they're alive. Have you never talked about prokaryotic cells at all? Did you not do it at all last semester? I thought you did. Prokaryotic versus eukaryotic, Venn diagrams. I think you made them.
We did not make a ton of others.
Okay, so tell me, what do prokaryotic cells have or not have? How about that? Okay, they do not have a nucleus. And we're going to broaden that even to they have no membrane-bound organelles. We always talk about the nucleus. They have no nuclear membrane. The nucleus is a membrane-bound organelle. But so are Golgi bodies, so are endoplasmic reticulum, so are lysosomes. So they have no membrane-bound organelles, but they do have, and this is bigger up there, maybe you can see it a little better. They do have ribosomes. Okay, ribosomes are not membrane-bound organelles. So they do have ribosomes. What do ribosomes do? They make proteins. So bacteria can make proteins because they have ribosomes. Remember, ribosomes are actually made up of RNA. So they're not membrane-bound, they're just made up of RNA. Yes, they are super teeny tiny. So you think your cells are small when you're looking at them under the microscope. They're about 10 times smaller than a eukaryotic cell. They can range from 0.5 to 5 micrometers. And an average eukaryotic cell is about 10 times bigger. There are some exceptions. There's some freakishly large bacteria. And there's some bacteria that are super small. But that's the general range.
When you say that the ribosomes are made of RNA, that's ribosomal RNA?
Yes,
Okay.
our RNA. Okay, how many cells are in a bacteria? How many cells are there? One. Okay, they're unicellular, of course, as it always is in science. There are exceptions in that some cells will actually remain attached to each other after cell division, and we'll talk a little bit later in the chapter about biofilms, about how they will associate with each other and kind of specialize, but they're unicellular. Okay, when we say they're small and they're unicellular, this isn't to imply that they're not organized. They're really highly organized. They have what they need to carry out all the functions of life. Okay, so when we talk about characteristics of life, they can reproduce, they can metabolize, they can get rid of waste, they can reproduce, they can grow, they can do all of those things, and all of that is packed into one little teeny tiny cell. That can do all kinds of other crazy things, like growing in weird pHs and salinities and radiation and surviving all kinds of conditions. So we sometimes think of them as like primitive because we think of them as simple. They're not simple. They have a lot of complexity and they can do a lot of things. You can see here they have different shapes to them, which we've been talking about in lab. Spherical, what's the word for spherical that we use? Coxide. No, a sphere and a circle are different. One is 2D, one is 3D. That is. Rod-shaped ones are called what? Bacilli. We're working with E. coli in lab this week. It's a bacilli. And then spirals come in there's various shapes of spirals. That's showing you a really cool one. Most of them don't look that cool. Sometimes they're put into the category of spirilla. Okay, you will notice in this picture, all bacteria also have a cell wall, which I tried to underline but kind of crossed out. And they also have a plasma membrane, which is similar. I just also crossed that one out. That's similar in function and structure to a eukaryotic cell membrane, which we'll talk about a little bit more on Friday. So they have a cell wall, they have a plasma membrane, they have ribosomes, they have a cytoplasm. We're going to talk about plasmids and fimbriae, and capsules, and flagella, and nucleoid containing DNA. So if we focus on the surface of the cell, I want the picture, sorry. I want the picture. This is prokaryotic. We are not touching eukaryotic cells until next week. So everything that we're talking about right now applies to bacteria for sure, and some of it to archaea. We'll talk a little bit more on Friday about the difference between archaea and bacteria. So the plasma membrane should look similar to you. What are these guys? Phospholipids, the phosphate head and the fatty acid tails. There's proteins embedded in the membrane. It's 2 layers. It looks a lot like a eukaryotic cell membrane. Then you can see these 2 different kinds of bacteria, and this is what we're going to finish up with. There's 2 different kinds of cell walls that you see here. And we're really into this when we talk about bacteria, because that cell wall structure can tell us something about the bacteria. This bacteria is gram-positive on top. The one on the bottom is gram-negative. So you can see the cell membrane. What do you notice about the cell wall? So there's a different arrangement in the gram-negative than the gram-positive. The gram-negative actually has 2 membrane layers. What else do you see? What do you notice about this cell wall versus this cell wall? The gram-positive cell wall is a lot thicker. Cell walls in bacteria are made of peptidoglycan. That's important because that's different than the cell walls of other organisms. So plant cell walls are cellulose. Fungi cell walls are chitin. Human cell walls we don't have them. Okay, and that structure of the cell wall is important for the way that we treat bacterial illnesses, even though most bacteria are good. I'm going to keep throwing that in there. So we'll talk more about that. I will see some of you in lab at 1 o'clock with your pre-lab. done, and we'll have a quiz.